Hi, my name is Jonathan Parr. I'm an assistant professor at the University of North Carolina at Chapel Hill in the Division of Infectious Diseases. I have had the pleasure of co-directing our division's inpatient COVID services since early in the pandemic. Today, I will be talking to you about the role of tocilizumab in COVID-19 pneumonia. First, a couple disclosures, competing interests I've listed here. None of these are related to COVID-19. However, this talk will include discussion of off-label use of therapeutics. Neither tocilizumab nor many of the other immunomodulatory agents discussed herein are FDA approved for use in COVID-19. So where are we going with this talk? First, my goal is to have all of you take a good close look at this picture. This is, in fact, a traffic robot from Kinshasa, Democratic Republic of the Congo. I've included it for two reasons. One is to remind me to say that in my normal life, I study the molecular epidemiology of malaria and other infectious diseases, mainly in the Democratic Republic of the Congo. So probably like many of you, I have found myself figuring out a new reality during COVID. My work in the Congo continues, but suddenly here I am giving a talk about immune modulators and respiratory diseases. I am not an immunologist by training, but I am a clinician who's been struggling, like many of you, to make sense of the evolving literature. The other reason I include this picture is that I think traffic robots are really interesting and are unique to the Congo. Instead of simply changing from red to green, like a normal traffic light, these robots actually rotate. So fortunately for you, this is the last discussion of traffic robots that I will include in this talk. And now let's get on with it. So what will we cover today? First, I'll talk a little bit about COVID-19 and the paradigm of cytokine storm. Then we'll focus in on tocilizumab and the evidence for and against its use in COVID-19. And finally, I'll attempt to bring everything together and share some lessons learned, not only for tocilizumab, but more broadly for decision-making during the pandemic. So as all of you know, the COVID-19 pandemic is global. Case counts have been rising across the world. This is not only true in the settings where you are practicing or living, but certainly true in the United States where I practice, as depicted by the map here. I've entitled this slide, No Rest for the Weary, because at least our experience in North Carolina has been one of kind of a steady stream of COVID cases throughout the pandemic. This is the entryway to one of our isolation units at UNC where I practice. And it has been a very challenging time, I think, for all of us. And for me, when I step through those doors and see a patient suffering from COVID-19, I am forced to ask lots of hard questions about what the right management choices are for that individual patient. So one question that I found myself asking regularly early in the pandemic and still now is why do some patients deteriorate so quickly? There are obviously lots of potential explanations for this, but it is certainly true that a subset of patients have a hyperinflammatory state. This hyperinflammation has been observed not only in SARS-CoV-2, but also in the original SARS-CoV-1 pandemic. This is a paper from about 15 years ago in which the term cytokine storm was used to describe patients who had elevated cytokine and chemokine profiles compared to normal. The cytokine storm paradigm has been rapidly adopted and was adopted quite early in the pandemic for a few reasons. One is that early observational data from China did show elevations of a subset of cytokines and of IL-6 in particular, and found associations with death and severe disease. However, independent of this evidence, I think the real reason that cytokine storm, the term, caught our imagination is that all of us in our medical practice and then also our personal lives truly felt like a storm had swept through and disrupted everything we thought we knew about reality. So when I walk onto a COVID unit and am seeing patients, one of the most difficult situations for me has been seeing that patient who's on 2 liters of oxygen in the morning, on 4 liters by 11 o'clock, and then suddenly, very quickly on a ventilator by mid-afternoon. Many of us have seen patients come in and have this very sudden, unexpected, and rapid decline, it's very logical to ask the question, should we intervene? I've included this picture of a pickup truck with an overheated engine, in part because I drove an old pickup truck as a teenager, and unfortunately, it broke down with some regularity. When I saw steam coming out of the engine, my usual approach was to pull over, turn off the truck, and pour in some antifreeze. That same approach, I believe, is clearly relevant in COVID-19. We've seen evidence of efficacy for dexamethasone in particular in reducing mortality in patients sick with COVID-19 on oxygen. That is not the topic of this talk, but I do think this analogy is worth considering because steroids, in my mind, are a broad anti-inflammatory and immune modulatory drug category that certainly affects specific cytokines, but also has multiple other effects. It's very logical to ask the question if perhaps we could be a little bit more parsimonious in our approach and use some of the newly developed monoclonal antibodies that enable targeted blockade, or to take the analogy a little bit further, allow us to cut specific circuits in the immune system engine that's overheated and make a difference. There is a pathophysiological basis for this assumption. This is a figure from a review published in Science early during the pandemic, exploring several potential pathways for a cytokine release syndrome in COVID-19. This particular schematic focuses on IL-6 and shows several of the drugs that have been considered for IL-6 blockade in COVID-19 pneumonia. I'm going to focus the rest of this talk on Tocilizumab, which is a monoclonal antibody which binds IL-6 receptors. It is FDA approved for use in rheumatoid arthritis and inflammatory arthritis, as well as giant cell arteritis. However, it is used routinely as well for treatment of cytokine release syndrome after CAR T-cell therapy. So let's take a moment to review the available evidence for and against Tocilizumab with the goal of answering the question, should we be using Tocilizumab in our COVID-19 pneumonia cases? Early non-randomized studies of IL-6 blockade were reported from China, and this particular paper by Zhu et al. Caught my attention. It was published in PNAS early in March and described a two-center experience with Tocilizumab. This is a retrospective study of 20 patients with severe or critical COVID-19. The figure here shows their key finding, which is that in this small cohort, patients appeared to have improvement in CRP and other inflammatory markers, as well as improvement in their oxygenation and their temperature. Similar observational studies came out from China and then also later from Italy showing similar results. In the second paper shown here by Luo et al., this is a single center retrospective study of 15 patients with moderate to critical COVID-19. The results were not quite as convincing. However, the authors still concluded that Tocilizumab appears to be effective in critical COVID-19. There was a larger prospective observational study from Italy published shortly thereafter involving 63 hospitalized patients with severe COVID-19, and again found improvement in inflammatory markers and oxygenation. So at UNC, where I was practicing, as I mentioned before, we were seeing patients come in sick with COVID and definitely seeing a subset of patients who rapidly deteriorated. We were very motivated to find solutions for these patients and did use Tocilizumab off-label for a small number of patients early in the pandemic. However, we were a little bit uncomfortable using such limited retrospective data to guide our decision-making. And so with the help of a very talented group of MD PhD students and a fellow who had been sidelined by COVID, we were able to do a very quick descriptive analysis of our first 11 patients treated with Tocilizumab. As expected, we did see rapid improvements in CRP after treatment with Tocilizumab, as shown here. However, we did not see the same clear evidence of improvement in other markers, such as oxygen requirements, temperature, or other clinical data. This was enough for us to step back, reevaluate the evidence, and make the decision to wait on further Tocilizumab use, except in very specific circumstances, we decided to wait for better data before including it in our treatment algorithms. However, shortly thereafter, there was just an explosion of observational studies on tocilizumab and COVID-19. This is a nice meta-analysis from Clinical Infectious Diseases published last month. The authors reviewed over 700 papers describing off-label tocilizumab use in patients with COVID-19. They focused in on the 10 highest quality studies, and as this forest plot shows, did find evidence of a mortality benefit with 12% lower mortality in tocilizumab treated patients. More recently, in mid-October, there was yet another explosion of important studies on tocilizumab that came out. And this study is the first that caught my attention by Gupta et al. This comes from the Stop COVID investigators. This is an observational study of 68 sites across the United States. These are leading academic centers with names that many of you would recognize. This particular observational study included almost 4,500 critically ill patients in ICUs and 419 patients treated with tocilizumab. Using inverse probability weighting methods and a simulated trial design with observational data, the investigators concluded that tocilizumab appeared to be efficacious in its two primary thresholds for efficacy, time to death and 30-day mortality. I want to also take just a moment to look at the 30-day mortality numbers, as I think they are a really nice metric that enable comparison across studies that were performed in different places and that were performed at different times during the pandemic. In this particular study of critically ill patients, the overall mortality rate centered around 30%. And you can see, again, that there did appear to be a mortality benefit in this cohort. However, in the same issue of JAMA Internal Medicine, the first two randomized trials of tocilizumab were published. I would like to take a few moments just to talk about the randomized trials available to date. As I mentioned, there were two open-label, non-placebo controlled randomized trials of tocilizumab, one in Italy and one in France. Shortly before, there were two important press releases released by Roche, tocilizumab's manufacturer, describing initial outcomes from two large randomized controlled trials. And then finally, the day after these studies came out in JAMA Internal Medicine, there was a robust, double-blinded, placebo-controlled, randomized controlled trial published in the New England Journal. So I'm going to take a moment just to review the high points from each of these studies with the goal of synthesizing this information in a way that allows us to inform our practice. So the first study from Italy involved 24 sites, 126 subjects with severe COVID-19. These patients did require oxygen, low or high flow nasal cannula, but they were not on mechanical ventilation or non-invasive ventilation. This study, unfortunately, was stopped early by the DSMB for futility. You can see key figures here at left. These Kaplan-Meier curves don't show much split between these two different groups. And in fact, the predefined thresholds for efficacy were not met. This is a composite endpoint here for improvement in, worsening in oxygen status, ICU admission, or death. And again, even though it wasn't a primary outcome, I think it is useful to look at the 28-day mortality here. There was no difference between tocilizumab treated and usual care treated patients. But most importantly, this mortality rate was very low and surprisingly low, given that this occurred in Italy early in the pandemic when mortality rates far exceeded this 2 to 3% range. I think one reason for this particularly low mortality rate is that in Italy at this time, certain patients were not eligible for ICU admission due to their age or other comorbidities, and these same patients were not eligible for the study. There was a very similar study conducted in France at 9 sites involving 131 subjects with severe COVID-19, a similar patient population in terms of oxygen requirements to the Italian study. However, these investigators restricted their study only to subjects requiring oxygen by low flow nasal cannula. You can see in the key figures here at left that unlike the prior study, there does appear to be some separation between these two groups. And in fact, one of two predefined thresholds for efficacy was met. Their early outcome of increase in clinical severity score at day 4 was not observed. However, there did appear to be an improvement in survival without non-invasive ventilation or mechanical ventilation at day 14. Looking again at 28-day mortality, you see that the mortality rate is more in line with what is seen in other sites like where I practice in the US, but again, no clear mortality benefit in those treated with tocilizumab versus those who received usual care. So stepping back now and looking at 2 important press releases from Roche. The first described results of the COVACTA study. This included 67 sites across North America and Europe, involved 450 subjects with severe and critical COVID-19. A good proportion of these subjects actually were critically ill, which is an important distinction as we think about how to generalize these findings. Unlike the prior 2 randomized studies from France and Italy, this multi-site study was double-blinded and placebo-controlled. It was conducted by Roche, which is tocilizumab's manufacturer. Their primary outcome was a difference in clinical status using a 7-category scale at day 28. You can see in the figure at left that there aren't marked differences between these 2 populations, and indeed the threshold for efficacy was not met. Looking again at the 28-day mortality question, there was no clear difference between these 2 groups. Mortality rates were around 19–20%. However, shortly thereafter, Roche released results from another study called Impacta. This study involved 69 sites across the Americas and Africa and enrolled 389 subjects with severe COVID-19. Interestingly, this study focused on what they describe as minority racial and ethnic groups, including predominantly Latinx communities, but also Black and African American communities as well. The majority of these subjects received steroids and also remdesivir. Subjects in the COVACTA study that I just mentioned, a portion of them did receive steroids and remdesivir as well. However, the study was conducted earlier in the pandemic and only a minority did for that study. So Impacta is different from COVACTA in not only its study sites and patient population, but also in the other COVID-directed therapies that patients received. This was a double-blind, placebo-controlled study also conducted by Roche, tocilizumab's manufacturer. You can see in the key figure here at left that there does appear to be a difference between tocilizumab treated and placebo treated patients in terms of the proportion with death or those who required mechanical ventilation at day 28, which was indeed the predefined threshold for efficacy. So that was met in this study. However, again, stepping back and looking at 28-day mortality, there was no difference between the 2 groups. And perhaps even a trend towards increased mortality in subjects who got tocilizumab. So I had the opportunity to review some of these studies for JAMA Internal Medicine and to try to put them together into a digestible format with the task of really thinking about how we could operationalize these findings. If you're interested in the details, you can find the paper through the citation below, but I'm going to focus in on the key findings, which are the results from randomized trials. In the figure depicted here, I'm showing primary outcomes across all studies. We've discussed all of these already in the previous slides, but in summary, 3 of 5 primary outcomes did not show evidence of efficacy. And among the 2 that did meet the threshold for efficacy, one of them just barely met the predefined threshold, coming in at 0.05% above the threshold. Looking again at 28-day overall mortality, there was no clear difference between these groups. One could rightly argue that none of these studies were powered to detect a difference in overall mortality. Nonetheless, the consistency of this finding across studies does suggest that there may be something to this story with no mortality benefit. So my conclusion in reviewing these studies was that the available data and results from randomized trials, while mixed, did not provide support for routine tocilizumab use in COVID-19 pneumonia. Fortunately, the following day, a double-blind, placebo-controlled randomized trial was published in the New England Journal. This came from the Back Bay investigators that come from 7 Boston area hospitals, including Mass General and Brigham and Women's. This was sponsored by Roche and Genentech, although was conducted independently by the investigators at these academic institutions. You can see the key figure here at left, no clear difference between groups in the Kaplan-Meier curve, and indeed the predefined thresholds for efficacy were not met. No difference in time to intubation or death, and looking at a tertiary outcome of 28-day mortality, there did not appear to be a difference between these two groups. So what should we do with this information? How should it motivate our decision-making about immune-based therapies? And also, are there lessons that we can learn from this process that are relevant to COVID-19 management in general? So as I mentioned before, it's clear that there is a role for immune-based therapy in COVID-19. The studies that are out so far looking at dexamethasone in particular for COVID-19 show evidence of a mortality benefit. This is primarily resulting from the large pragmatic recovery trial being conducted in the UK, but also supported by several other smaller trials that were summarized in the meta-analysis I featured here. However, while there is clearly a role for immune-based therapies, and dexamethasone is now the standard of care worldwide for COVID-19 patients on oxygen, the role of targeted immune blockade remains unclear. I have given a few examples here that I think make this point nicely. Anakinra is one drug that got a lot of attention due to an increasing number of positive results from observational studies. This storyline should feel somewhat familiar. But just last week, there was a news release from one of the regulatory agencies in France announcing that the large Anaconda randomized trial had been stopped due to evidence of excess mortality in the Anakinra treated group. So quite a concerning finding. Additionally, Cerulumab is another IL-6 receptor blocker similar to Tocilizumab. We have a press release from Sanofi saying that the randomized controlled trial of this particular drug failed to meet its thresholds for efficacy, and that the company does not plan additional COVID-19 related investigations for that drug. However, there is a long list of other anti-cytokine and chemokine agents under development and studies that are underway, including some of those listed here. It's entirely possible that we may find a drug or a niche in which these drugs are actually useful, but at least at this point, from my standpoint, we don't have strong enough evidence to support their routine use. And that takes me to what I think is the most important point from this venture for me, which is now is the time to rely upon randomized trials. Early in the pandemic, all of us were struggling to find treatments of any type that we could use to help patients who were rapidly deteriorating in front of us in the hospital. And it was not completely unreasonable to use small studies with 10 patients or 20 patients to guide our initial treatment decisions. However, those days have come and gone. Now we have over 67,000 COVID-19 publications on PubMed. There are more than 2,500 COVID-19 clinical trials registered on clinicaltrials.gov. We can and should base our decisions on the best available evidence, which in my opinion comes directly from randomized trials. This viewpoint, I believe, is generally reflected in the current NIH guidance documents. I've pulled out one summary table for immune-based therapies here, and you can see that at least the US NIH right now recommends the use of dexamethasone with or without remdesivir, but by and large recommends against the use of most immunomodulators, including anti-cytokine drugs, outside of clinical trials. So in summary, a hyperinflammatory state can be seen in COVID-19. Whether cytokine storm is the right paradigm or not is another question that I didn't get into in this talk, but I'm of the mindset that it probably is not the correct paradigm. Observational studies supported rapid uptake of targeted immune-based therapies like Tocilizumab. However, randomized trials to date did not support their routine use and certainly did not support the routine use of Tocilizumab, in my opinion. Additional RCT results are expected, including those studying different patient populations, including studies involving more critically ill patients. So we may see a different storyline in the future, but with the evidence that we have as of now, we do not plan to add Tocilizumab to our treatment algorithms at UNC anytime in the near term. And last but not least, data from high quality randomized controlled trials is essential to identify therapeutics with real impact. Thank you for your attention. If you are interested in the work of our research group, again, in my normal life, I focus on molecular epidemiology of malaria, syphilis, viral hepatitis in places like the Congo. Happy to talk more about that, or you can find me on Twitter. I'm a new user, but enjoying using it to find evidence about COVID and other new therapies. Thank you very much.